Welcome everyone to our August webinar. My name is Matt Del Bono, an Extension Support Specialist with the Glaze Consortium. And today we have a presentation from Dr. Rhuanito Ferrarezi from the University of Georgia on adapting fertilizer management to enhance plant nutrition in greenhouses and vertical farms. Before we jump into his presentation, I'm going to welcome you all here today with just a very brief introduction to our organization, Glaze. So Glaze is the Greenhouse Lighting and Systems Engineering Consortium out of Cornell University. And we are here to transform greenhouse lighting and systems management to support climate CEA, climate smart CEA. That is what we are all about. And so we do so by conducting research and educating growers so that emerging technologies can be adopted faster and help growers save money and reduce the environmental impact of energy use in greenhouses. We do our work in a number of ways. We do so by conducting research. As I mentioned, much of our research is focused on greenhouses, but also indoor agriculture, and we seek to help technology transfer through showcasing the results of new technologies' impact on plants. We have webinars, we have our annual short course, and a variety of other education and workforce development initiatives that our members take advantage of. And we also find that our members value our networking benefits via our in-person industry advisory board meetings, as well as our online meetings, and of course, our annual summit, which I will tell you a bit more about in a moment. Our members are highly valued, and we hope that you consider joining Glaze. And if you are interested and would like to learn more, I have our membership categories on our screen, and we would be happy to connect with you offline and talk more about our organization. The last two quick things I want to plug before I hand things over is that this year we have a new membership initiative called the Vivid Canopy, where folks working in CEA can connect and organizations can showcase how they are leading by example and hiring a more diverse workforce in CEA. And at the end of the month, we're actually hosting a Vivid Canopy breakfast panel at the CE Ag World Conference and Expo in Raleigh, North Carolina on August 28th. So please reach out to me if you're interested in registering for that event. We certainly hope to see you there. And finally, I want to encourage everyone to consider joining us for our annual Glaze Summit in Montreal on November 6th. We have a very exciting agenda shaping up, including a keynote presentation from Dr. Tessa Pocock, Chief Science Officer at Solely Organic. We have a US and Canadian grower panel coming together, a research panel, as well as a number of other presentations on new technologies. Our theme is dynamic lighting, and we hope to see you there. I also want to thank our anchor partner, Saloon, who's helping make this event possible, and of course, Hogendorn as well for sponsoring our networking event. So today we are here to learn about fertilizer management, and I have the pleasure of very briefly introducing our speaker today. Dr. Rhuanito Ferrarezi is an associate professor of controlled environment agriculture, crop physiology and production laboratory at the University of Georgia. His program focuses on precision irrigation, plant nutrition, crop physiology, biopharmaceuticals, lighting, imaging, and cropping systems for greenhouses. And I know you also have a brief intro slide as well. So I think I will keep it brief and stop my share and invite you to take things over. While you're pulling up your presentation, I'll just remind our audience today that we will have time for questions at the end of the presentation. And so please feel free to submit those as we go. We will work through them in the way that they came in at the end of the presentation. So Doctor, please feel free to take it away whenever you're ready.
Thank you, Matthew. Appreciate it. Good afternoon, everyone. That is on Easter time. So I know some of you might be all over the place in the States and abroad as well. So I saw a familiar name, Mahdi Faraji, with us today. So thanks for sparing time to hear a little bit about one of my projects. I would like to use this opportunity to thank you, Matthew, to coordinate this with me, and also, the Glaze leadership through Gretchen inviting me for this webinar. I've been interacting with Glaze and people involved with, this organization for a long time, and it is my pleasure today to come over and talk a little bit about one of my signature projects that just completed through a master's student that graduated this summer. So as you can see over here, I have a list of co-authors, and the first name is Chris Nieters, who is now doing a PhD at the University of Minnesota, and several other colleagues that are listed over here as well. So we are going to focus one area um, of my program. So I just wanted to tell you that I've done all my training in Brazil for the ones that don't know me, and had been around UGA for a long time, combined for 5 years, even though I had experiences in between at the University of Florida and the University of the Virgin Islands. So as Matthew highlighted, my program focuses in crop physiology and production. So I've been doing quite a lot lately with the merging of two labs into my program. So if you are interested in seeing what I do, just go ahead, Google Ferrarezi lab.uga.edu or scan these particular QR code, and you are going to see the scope of my work that had been published lately. So I have a very vibrant and strong research program, but I also do teaching. So teaching is one of my core missions here. You are going to see, basically I have a twofold mission: research and workforce development. So my activities are related to undergraduate and graduate teaching and research. And we do have a bunch of people around the lab that, work hard to get the data that we need to disseminate to you all, growers, researchers, stakeholders, industry members. So I do have a lot to talk about concerning my programmatic areas, but today we're going to focus in plant nutrition. So I just have a few slides to bring you up to speed on the topic. So you can see at this very nice figure that there are multiple elements that are on the circles over here that are related to plant growth and development. And they all influence certain parameters in plant growth and development that are key for physiology and consequently growth. And we are all interested in yield and quality, so those nutrients play a key role, and that is going to be the target of today's talk. Just wanted to bring something up to the table here, that is the close interaction between pH and nutrient availability. On the left side here, we have how soil pH influence nutrients, and on the right, we have how water or substrate-based media influence pH. So we will target a lot of fertilizer solution management strategies, and that is intimate related with pH. So whenever you all observe that plants are having problems, so that is usually related to what we see here in the top. Reduced growth, issues with colors that are sometimes associated with deficiency symptoms. Sorry. And of course, we have a whole array of pest diseases and other issues. But most of the times, what we are targeting in this particular conversation are related to nutrient deficiency related symptoms. And we have a lot of tools in our toolbox to achieve a proper plant management. And in green over here, you can clearly see that we have a lot of possibilities concerning analysis: foliar, substrate, water, sap analysis, which, by the way, is an important topic of research at my lab, and also visual diagnostic. So we then can sample and get those analyzed by labs, And what usually happens is this cascade of events that are really related to ways to track how our plants are growing. So that is like a blood test for our plants, right? So all of these is either done on schedule by the growers or by demand. And we all know these is super expensive, as highlighted here by this, steak wrapped up in gold, right? So this is a core activity that growers, researchers should do. But at the end of the day, due to the cost and the labor associated with it, there is a tendency to basically flush, the reservoirs, discarding those solutions, and then refilling. So research data shows around the world, by multiple operation sizes that a lot of um, fertilizer is discarded. I forgot to start my timer here. So the frequency of discard is really high among growers, and that, as you know, has multiple impacts to the environment related to nutrient load in water bodies, particularly causing eutrophication, and also a massive cost because these nutrients are dumped instead of being recycled. There are reasons for this to be done, so related to pest, disease, challenges, managing the solutions, but overall that is done because it's much simpler than any other method. So to tackle this, so nowadays we are all using supplemental lighting or sole source lighting to grow plants. So that being emission of the glaze itself, congregating a lot of people that represent this industry. So I brought these over just to highlight that on the left we have the effects of light intensity, and on the right the effects of light quality on plant production. So those parameters are usually considered separately. So we do have them one problem that I want to tackle today. So what is the impact of the nutrient solution refill strategies on yield and health of plants that are growing under increasing light levels? And going beyond that, so do production systems like greenhouses and vertical farms differ in how plants perform on that? So we are then also interested in how the fertilizer use efficiency differs when we have those two conditions analyzed. So this came out from multiple requests from growers all over the state of Georgia, and several vertical farm operators, two of them that I collaborate closely. And we came up with this project, really looking to compare the performance of 6 targeted strategies to grow lettuce and spinach under 3 lighting levels in a greenhouse and a vertical farm. So as you can see, so it's a very ambitious objective, and I delve into it. So here you can see a picture of our greenhouse and a picture of our vertical farm. You will see a few other pictures along this talk. So as you can see here, maturing methods will be the same for the 2 crops that we grew, and then we will separate it out the results and discussion by crop. But we had then repeated sequentially one after another. And let me start by telling you what we used as nutrient solution refill treatments. So on the left over here, please bear with me. So this is one of the most important parts when I'm going to tell you what each treatment is about. So number 1, we have the drain and refill treatment. Number 2, we have the top off of a full strength solution, meaning that we are using the same solution that we used at the beginning. We had another treatment where we have the strength of the solution to save fertilizers. We had one of the industry standard treatments, so we used water to refill the tanks and then those to a target EC. One of the most different treatments is the water use efficiency. I will explain to you in a second, and the individual nutrient dosing. So from left to right, we have a non-targeted to a highly targeted refill set of treatments, indicating them several procedures, such as, for example, the water use efficiency. So that potential treatment is based on the weekly water use efficiency of the plants that are growing on a given system. So we adapt what we provide based on plant growth and development. So we compare whatever we have as a target for the tissue element, and then we calculate the water use efficiency of the refill solution, really highlighting the amount of nutrients that should be added to sustain plant growth in different phases. All right, so we based these from a recent publication from Lingenfeld in Bugby. So moving on, so we had then 6 different NSR treatments, and we had 3 lighting treatments. So as you can see here in the top, we started with the standard 250 micromoles and increased it to 500 and 750. Just a brief disclaimer to everyone. So we quite frankly did not get 750, we got 700, but regardless, so pretty high light levels. So we had a fixed photo period. So those are the DLIs, as you can see, from 14 to 40 moles per square meter per day. The spectrum distribution was the same across treatments. Just wanted to show you a picture of our vertical farm. So this was totally built in-house by the student. So such a talented student that put all of this together, all of the wiring, all of the equipment decisions. So we joke in the lab that these student actually got trained as a crop physiologist, but also as an electrician, as you can see over here. So that is not trivial. And I acknowledge, all of the great work that Chris did. And basically, due to several challenges, I just wanted to bring up a phrase to everyone, so we have to go a lot of wrong roads to find the right one, right? So we use the same idea behind this particular treatment in the greenhouse. Here's a picture of the greenhouse. On the right here, we see 4 light fixtures, 8 and 13 light fixtures to achieve the light levels that we wanted. And just wanted to say that we use the same photoperiod, DLI, and spectral distribution. So we just measured all the environmental parameters, so I'm not going to spend too much time on them, but just to highlight that when we had the greenhouse, we added up the solar radiation because that was polycarbonate greenhouse. Our treatments were applied across the board on a design that was a randomized complete block design, 3x6 factorial. Had 3 replications. Each of our trays had 4 plants, as you can see here on this styrofoam. And then we had a tray where we had about 24 liters of solution. So we tested 2 different crops, as I mentioned, and those 2 cultivars were the ones that we chose, Green Forest for lettuce and Sun and Gel for spinach. We collected a lot of data. You are going to see a few of them. So we had data collected daily, weekly, and during 2 different time points. So for the sake of this presentation, we will focus on the most important data, the ones that really can bring something to your day-to-day. The environmental conditions, they varied. Inside the greenhouse, for example, we did not have CO2 control because the greenhouse was an open system with a pad fan cooling system, while in the vertical farm we had a high CO2 level and the VPD changed as well in the two environments for that reason. All right, so are you ready for the most important part, the results? So let's begin with lettuce. So the graphs will be similar. Okay, so this one in particular is split in three different parts. So we have in the top, well, in all of them, fresh canopy biomass, and in the top we have the production systems. In the middle we have the different light levels, and in the bottom the six different nutrient solution refuel strategies. As expected, vertical farms, or in our case, the vertical farm yielded a higher biomass than the greenhouse. You can see here at the center that the fresh canopy biomass increased as we increased light levels. So nothing new in there. And what surprised us was at the bottom here that, let's look at the data on the right side, that is at the end of the plant growth cycle at 28 days. So there was no difference concerning plant growth expressed by canopy biomass among the different treatments that we tested. So that's great news. So that's basically, one of our major outcomes of this project. So over here on the left, we have the greenhouse data, and on the right, we have the vertical farm data, and we have chlorosis in the top and tip burn in the bottom. As we increase the light intensity of our treatments as expressed here in the bottom, we did have an increase in these symptoms, particularly tip burn, as expected due to the increased transpiration. All right, so that was way more proeminent in the vertical farm due to the environmental conditions and the stress that the plants were subjected in that environment. When we look it up at these two particular parameters, so basically there was no difference. When we look at the bottom here, the nutrient solution management strategies did not have a significant impact on chlorosis and tip burn. When we look over into some parameters to help explain why we obtained certain results. So leaf pigment is one of the most important ones. So we did have a difference for chlorophyll content concerning the environments that we tested. So as you can see here, with the increasing light levels, we had increased leaf chlorophyll content. And concerning anthocyanin, so higher the light level, higher the anthocyanin content, indicating, a stress response for those high light levels due to the photo inhibition and the stress that plants were suffering. Our experiment measure then, as you can see here on the left, so the tables will be very similar to each other. We will have all the three factors that we tested: the production system, the light intensities, and the NSR treatments. Let's look at the end of the cycle here. So concerning water replenished, so we can see that there was a major difference between the amount of water added in the treatments that we tested. So these really shows, for example, for the nutrient solution refuel strategies that we used, that the system that drained and refuel used the most, while the other ones were very similar. That has a major impact on something that we will see in a minute. Concerning the fertilizer replenished, we also had the same trend here, which is expected because we are dumping and refueling. We discussed a lot how we would express fertilizer use on these experiments. So we came up with one simple layaway to indicate that is the total amount of fertilizer added. We will in a minute show the fertilizer use efficiency, but this was just a way for us, for example, to indicate, for each of the 6 treatments that we tested, which one used the most nutrients or fertilizers, right? So in red over here, you can see the drain and refill, and then, it follows up the targeted approaches that we selected. So you can clearly see a huge gap between the treatments. So overall, we used 80 or more, 80 or less percent fertilizer when we used a targeted refuel strategy as opposed to draining. And that is impressive because despite of us knowing that is the case, so we don't have studies indicating how much we would save when choosing better strategies. So that had, of course, a direct consequence on the individual nutrient use efficiency. As you can see here, we only decided to express 3 nutrients, nitrogen, phosphorus, and potassium. And what I would like to bring to your attention is the fact that as we have a higher water disposal with fertilizer, so then we have a much lower nutrient use efficiency. As we go to a highly targeted refuel strategy, then of course we are increasing the fertilizer use efficiency, which is our goal with this strategy, validating our hypothesis. Another important aspect, so at the beginning I talked about pH, so you can clearly see here that when we sum all the pH amendments used, so the treatment in the vertical farm that had the drain and refill used the majority of the pH amendments to guarantee that pH was stable in the level we wanted, indicating that this not only wastes water and fertilizers, but is also difficult to manage. So I am making the data available to Matthew, so he will share by demand the slides. So we did go very deep into the understanding of tissue, solution, nutrient concentration. What I would like to bring to your attention is that despite of changes that we had concerning the tissue nutrient concentration, we didn't have anything that was really different than what we expect in this kind of research. So basically our levels fell within the expected range with a few exceptions, and this is an old recommendation. We don't have many of them available. What I would like to show it to you, nothing caught up our eyes with the exception of copper, that over here in the bottom we have the nutrient solution refuel strategies, and in the gray bars here we have the increased light levels. So in the greenhouse the levels increased with the increase in light levels, and in the vertical farm those concentrations decreased over time. So quite interesting result over there. So we then tested the solution nutrient concentration, and we will have the NSR treatments here in the bottom for each of the graphs. But what I would like to bring it to your attention is the fact that the dotted line here represents the initial concentration, and then over time, at the end, plants were taking up those nutrients, really using them all. So what we saw for nitrogen and potassium was a decrease in the solution nutrient concentration with the more targeted solution, because we were maximizing the amount that they needed based on demand. That is different than just refueling based on a full strength or half strength, or even the drain and refill. So we found something similar for the micronutrients, really highlighting that we can guarantee that plants grow as well as we would want them, with less nutrients being, left behind in the solution. So that highlights the gain of this strategy. If we want to go complicated, so we did run Pearson correlation between the tissue concentration, as you can see here in the bottom, and the solution concentration, as you can see here in the top. So I think the overall message that I wanted to bring it to you, if you don't already know this. So what we have in red here represents high correlation. What is in blue represents a low correlation between these two nutrient contents. And when we have an X, that means that basically there was no correlation, right? So we can clearly see that the data is all over the place. Surprisingly, again, so you can see here in blue, copper in solution had a negative correlation with pretty much all of the tissue levels. All right, so just as a reminder, copper is very important for disease tolerance or resistance. So this is something to watch out when you have these strategies. So as I told you, we collected a lot of data. I just wanted to bring something up for your attention. So there is massive difference concerning gas exchange between between the production systems that we tested, which is highly expected, explaining, the responses that we had for these two production systems. So we also had an effect of light levels, what is also expected, but no major interactions with the nutrient strategies that we had, highlighting that picking a better strategy is not going to reduce plant performance. So to finish this part with lettuce, so we created a summary of all the factors that are a little challenging when we are managing solutions. So we started with the fertilizer, applied the pH drift, and all the other factors that you can see here, labor, cost, and risk. And we kind of ranked them based on the treatment that we chose to help you all understanding, for example, that we might have a high fertilizer applied in the drain and refill, but then the cost of labor of a more targeted solution is high. So you always have to balance those when you are making a decision to implement, a more targeted strategy. Meaning that then, of course, the ones at the middle here will have a more moderate cost compared to the one in the top. Sorry, compared to the one in the bottom. All right, I wish I could, interact with you all and see your faces and really see if you are interested, if there is something that I could answer, because now we have this spinach data to go over. All right, so just a disclaimer, since the experiments were set up the same, so they were just done in sequence, so the data presented is going to be the same. Sorry, needed to stop for a second to drink some water. So you can see over here that the tendons was pretty much the same, with one important observation. So when we look over the fresh canopy biomass of spinach, under increasing light levels, we did find a plateau on the 500 micromole treatment that equaled the 700. So really highlighting that it is crop specific, as we all know. We were really hoping to see an effect of lighting in NSR, but unfortunately that didn't hold. Basically highlighting that there is no change in yield based on this strategy. All right. So at the end, this is a great result because you can really help people to shift to a more targeted fertilizer management strategy without any negative impact in yield. We saw similar trends with higher tip burn in the vertical farm and higher values as we increase the light levels. That then was tied up to a little bit more visual chlorosis that could be seen in these particular conditions. So the chlorophyll and the anthocyanin data followed through what we were expecting. So since we are repeating, the same parameter, but with different crops. So plant physiology performs the same, despite of the conditions. So the first data that I showed to you, the biomass is the response data. All of these are control parameters to highlight then why those, why we had the response that we found in our plants, basically. So the data that I wanted to show to you here is a difference between, the production systems concerning water and fertilizer added. The light levels only varied concerning water replenishment. But then again, we had a massive difference between the amount of water and fertilizer added in the drain and refill in comparison to the other treatments that were saving water and really recirculating them. Again, that fertilizer use information for you, so hopefully this will stick and will really highlight that we do use a huge amount of fertilizer and we dumped into the environment when we use the strategy of dumping and refilling. So more or less same numbers, so roughly higher than 70% for the EC and over 94% reduction when we have a more targeted strategy. The same then happened with nutrient use efficiency. So when we are in the vertical farm, we use nutrients more efficiently. When we have the increased light levels, so 500, for nitrogen seems to be the right spot. And then when we look it up concerning the NSRs, so basically we do have an increase in these particular efficiencies as we go to a highly targeted approach, validating our hypothesis as well. So over here, I'm showing you the amount of pH amendments that were used. And I just wanted to highlight to everybody that it was slightly different. So we did use way more pH amendment on the EC-based treatment in spinach in comparison to lettuce, which was a surprise to us. All right, highlighting how sometimes, industry standard methods such as EC-based can be challenging to manage. So I will just make the same comment here. So we had almost no significant interaction for most of the tissue in the solution nutrient concentration. The majority were within range, or the amount that was out of range was not, causing any toxicity or deficiency. So we had pretty much the same effect as we moved, from a less to a highly targeted strategy. So we had higher nutrient used by the plant, having, a small amount being available in the solution, meaning that we were really optimizing the amount of nutrients that were being applied by the systems, or by the treatments in this case. Okay, so the same tendency for nitrogen, potassium, and the other two secondary macros, calcium and magnesium. So no significance at all for micronutrients. So look it up over here. We have in the bottom, Matthew.
Yeah, I was just going to mention quickly, as you requested, we're at the about the 40-minute mark. Sorry to
Well,
interrupt.
wonderful. You're almost there. Then I will leave plenty of time for questions. So this graph shows, the tissue concentration in the bottom, the solution concentration in the top, and this colorful map shows the correlation coefficient. Again, red means high correlation, blue means low correlation. So, and the axis indicates no significance, between these two factors. So we have negative correlations between tissue and solution concentration. So watch out then when you are planning your crop based on solution, or trying to interpret a status, because spinach then is way more challenging to manage, and everybody that grows that crop knows that. So another major difference between the other biometric parameters that we had here, I don't think I mentioned to you, but we have dry canopy, dry root, leaf area, specific leaf area, and then the physiological parameters that we use to determine gas exchange and quantum yield. So we had a much more efficient system in the vertical farm as expected. That's why everybody's super enthusiastic with it. We did have the same trend in terms of transpiration and no effect of physiology for spinach. And I would like to bring one detail to your attention, that despite of the significance in terms of fresh biomass, so we did find a negative effect of the treatment that basically dump and refill for spinach growth, which highlights, for the greenhouse and the vertical farm production, that this crop really needs to be studied a little deeper in warranting more research, as you will see in a second. So I did have that nice summary to highlight then the same parameters that I showed it for lettuce, really to help you navigate through the process of figuring out if the reduction, in fertilizer disposal can be offset by the cost that these more targeted techniques will have, and the long-term environmental concerns and impacts that are associated with, those strategies. Our conclusions are straightforward. Sorry. So we reduce the amount of water and fertilizer used by over 90% when we don't drain and refuel. So that is massive. So we basically have then the treatments that we tested, like half strength, water use efficiency, and the individual nutrient dosing, reducing the amount by 86%. So we need to tailor, these strategies by crop. And we noticed that these targeted strategies not impact plant yield or health of lettuce on both environments, but when we look at spinach in the greenhouse, so those targeted strategies, they did impact yield. The light intensity does not impact the performance of these strategies, but since plants grow faster, they transpire more, so it increases the rate of the nutrient solution replenishment as expected. So we definitely have an open field to do research to optimize plant growth under these broader systems approach that takes into consideration multiple factors that are related to plant growth. So with that, I would like to really thank the person that conducted all of these trials. Is the guy highlighted here in red, Chris Nieders. and wish him a successful PhD program under Dr. Nate Islands in Minnesota. And also, this huge, amazing group of people that work with me and had been making great progress as, in different fronts. We also have the strong support of USDA NIFA SCRI through the LAMP grant and several companies that are part of the glaze, such as Rain that I noticed, that really supported these initiatives. With that, I would like to thank for your patience. And I wanted to finish up taking your questions and also leaving my contact information here in case you need to reach out to me. Thank you very much.
Thank you so much for your presentation. And we do have several questions queued up here already in the Q&A, and folks continue to submit those as we've got 15 minutes here. The first question I'll read. This is short. I hope you if there's not enough information to know what it's referring to, we can encourage the person to provide more information. But at 2:17 p.m., just so you know when this came in, someone wrote fixed variable wave spectra with a question mark. Do we need a bit more information to know what they're referring to?
Probably just to indicate that we were able to vary the light level that we tested. So we had three light levels, and we kept the same, blue to red ratio, not changing. That was just a further explanation from my end to indicate that we only tested light intensity, not light quality in this study.
Okay. And Ed, if we didn't catch what you were hoping for us to touch on, please feel free to submit the question again. Another question that came in is, did you see any algae growth in DWC based on various nutrient dosing schemes?
Yes, a lot of it. Yeah, so that system is always challenging concerning, algae. And I think hydroponics as a whole concerning, lighting research because they have the ideal conditions. We did have it, we tried multiple approaches, industry standard approaches, cleaning using xerotol with various rates of I couldn't even say success, probably various rates of failure because they grew anyways, despite of, our attempts. There wasn't any, anecdotal visual observation that one treatment was better than another. Of course, the ones that, we dumped in refill, by definition will tend to have less algae growing because we dump.
Hmm, very good.
Would you like me to stop sharing my screen, or is that okay if I keep it there like this, Matt?
If you'd like your contact information up, that's perfectly fine. But also feel
Okay,
free to, feel. I think it's perfectly fine. Yeah.
okay.
Another question is, can you or have you used orthogonal statistical tests to rank the different NSRs per plant, per system, per lighting intensity, and then continued based on ranking by growth?
Excellent question. All of them were, but this one in particular, unfortunately, no. The, short answer. They'll be great to perform. This was a master's level research. I would say probably using orthogonal analysis, we would be able to see certain differences that, the standard stats didn't pick up. So maybe we are, in the process of submitting this to a journal, but perhaps if the person that asked the question is interested in having access to our data set and working with us to just depict that, I'm open to it.
Very good. So another question that came in is, how do you think the different nutrient solution treatments would differ over a longer period of time, maybe over several months or years after multiple crop cycles?
Good point there. So as you can imagine, so this was an experiment, right? So done for a short period of time. I am pretty sure that based on my experience with growers, that there are other factors that play a role, microbes in particular. So I would say that with targeted management, so we would tend to balance out the composition of the solution over time much better than just either adding solution at full or half strength or simply managing with EC. So large operations, they tend to keep solution steady. Of course, I would take it back. They tend to keep the solution for a long time. They just keep adding fertilizers. I would say the tendency is to have accumulation of several elements, while some will have to be added more frequently. The micronutrients being the most difficult ones to manage just because of the amount that plants uptake them.
Okay. This is a quick question. Did someone ask if you collaborate with the University of so UGA's extension services?
Um, Not officially, like through an agent in particular. By the time that we set this up, so there were no specialists, particularly professor in the college working with CA that had an extension appointment. So then we truly went directly and talked to growers, visited their facilities, and heard, how other colleagues, particularly from the vertical farm space, were managing their solutions and needed help with.
Very good.
So now we have a core group of UGA extension people. So we have three new professors with a heavy extension appointment, focusing in many different areas. So absolutely will be amazing to have those professionals when we started to do that particular collaboration.
Very good. We have another question that starts with a great presentation. So I wanted to share that.
Great.
And then
Thank
goes
you.
on to yes, and then goes on to say, you said the highest tip burn occurrence as light intensity increases is due to higher transpiration, but higher transpiration generally means higher CA uptake. In this case, shouldn't tip burn be lower with increasing light intensity?
Um, well, what I mean by that, then the overall conditions, they tend to be pushing faster plant growth. So there's more into that than, just the transpiration itself. So we increase transpiration. So then we definitely need higher airflow to guarantee air exchange for, the T-burn symptoms not to happen. So what I tried to convey there was the fact that basically with higher light levels, so the tendency to have tip burn increase just because the plant is demanding more water and nutrients, and we need to guarantee that the airflow that is required to avoid tip burn or to reduce tip burn occurrence is also provided. So we didn't vary that particular variable in our experiment, meaning that the low level in the high light level, so they had the same airflow at 1 meter per second.
Yeah. Thank you. We have another question. Did any of your NSR treatments lend to more observable deficiencies than other treatments?
Great question. Visual deficiencies? No. We noticed um, in that is tied up with the tip burn. So we noticed that treatments that had higher tip burn incidence had a little bit more chlorosis around the tip burn occurrence point that is associated. So we noticed that that is a dual parameter. People measure tip burn and chlorosis around that area as a potential indicator of tip burn occurrence. But no, we maintain the fertilizer levels or the nutrient levels at the adequate levels to guarantee that we did not have that effect.
Very good. So we have a question that maybe you can just give some guidance on or resources, but it reads, I know you cannot recommend any particular manufacturer, but in general, could you suggest a list of manufacturers of grow lights? I'm interested particularly in system design for small scale lettuce production using NFT channels. Any general feedback or there
I
or
Absolutely, I do have it. As you can see, so I have two lighting companies working with me. So one provided lights for my vertical farm, another for my greenhouse. So I do think that depending on your condition, so you would need to look at the reputation of the company, the ability of the technical people to assist you in designing your setup with the best return based on what you need. Also, the ability for you to scale, the ability for you to make changes along the way if you decide to change crops, durability, efficiency of that fixture that you select, also the durability, the um, customer service. So all of those are general rules of thumb because there are so many companies out there, some domestic, some international, some that just resell the fixture. So I do think as a rule of thumb, so you need to use the same caution as you use for any product that you buy, with the highlight that lights cost so much. So a little bit of research needs to be done before investing thousands of dollars in a facility.
very good. We've got just a couple more questions. I think we can get through them in the next few minutes. Someone ask which type of NSR disinfection was used, if any.
We did this, in a small scale. So it was large enough for us to conduct with our limitations, but there was no need for disinfestation during the experiment. So we had algae. Besides algae, so we did not disinfect our solution against any disease using any technique, which of course in a commercial production facility is slightly different, right? So we can have active and passive systems, which we did not use any.
Thank you. And I'm just trying to get in these last couple questions here. For each NSR strategy, did you tally the total volume to drain per test? Is that data available?
We did, yeah, we tally everything. So I did provide that for both crops, and I can really make it available, in details if you need it, for sure.
Hmm.
Yeah, there was a table on each of them, one of the first tables that highlighted the amount used.
Very good.
They vary so drastically, usually tenfold from the dump and refill, and that goes hand to hand, water and fertilizer.
Thank you. I think we've got time for one more, and I think we've got them all here. Someone asked, what nutrient strategy would you recommend for a large scale production VF or mix of strategies to reduce accumulation, depletion of nutrients?
Great question. So I would say, as you could see, so there was no difference between the strategies. Okay, so that means that is an awesome outcome because we can go and pick the one that best suit our needs. For example, the addition of the half strength solution is a great one because we add a weaker solution, but then we will accumulate over time certain elements that plants are not taking up. The use of the water use efficiency or the lab method are, to me, the best ones because they take first the plant needs and plant growth into consideration, and also, the nutrients that are available in solution respectively. So, however, they will require a certain consistency. They will require you measuring plant growth over time for the first method, or really collecting samples, sending it over to a lab, being on top of when they come back. Have to have some sort of agreement with the lab to send you the samples, back ASAP for you to make an adjustment. So I think, that being said, those three separately or hopefully in combo, so we will allow you to really dial down the amount of fertilizer added.
Very good. Well, it is just past 3 p.m. So we do I think this is a good stopping point for us. I want to just wrap things up by saying thank you so much, Dr. Ferrarezi, for this presentation. Clearly, it was very interesting to our audience. We had a lot of great questions, and so we really appreciate your time and participation in our webinar.
Thank you very much, Matthew, and thank you all for being here today. So I believe that this is an open possibility for me to assist you individually. So please don't hesitate in reaching out to me if you see the need. I will be glad to help.
Thank you so much, and yes, I'll just wrap things up by saying we appreciate everybody's time to learn more about this presentation and the Glaze Consortium, and please feel free to contact me if you have any questions. And this presentation was recorded, so we will make that available on our website for anyone who wants to go back and review, and also to keep an eye out on our events calendar. We have other webinars scheduled as well as our in-person summit in November. So thank you again, everyone, and I hope you have an excellent rest of your day. Thanks.
Thank you all.